Now, please let me introduce the second speaker, Dr. Ying Li, Director of Purification Process Development Department. Dr. Ying Li leads the CMC purification process development and provides technical leadership in new technology platform development. She joined GenScript ProBio in 2022, bringing over 10 years of biotechnology experience spanning from multidisciplinary areas. Prior to joining GenScript ProBio, Dr. Li was an R&D manager in Merck KGaA from 2017 to 2022, led the development of viral vector manufacturing platform and the biocontinuum platform. Dr. Li obtained her BS and PhD in chemical engineering from National University of Singapore and the Pennsylvania State University, respectively. She is a frequent speaker at ACS and AICHE and has authored 8 publications in top-tier bioprocessing journals. In this talk, Dr. Li will be discussing development and manufacturing strategies for antibody-drug conjugates. Now, I'm delighted to hand over to today's speaker, Dr. Li.
Right. Thank you very much, Jessica, for your introduction.
You're welcome.
Going to share. I hope you can see my screen well.
Yeah, it's great. You can start now.
Right. Right. Good afternoon, everyone. It's my great pleasure to present today's topic on the development and manufacturing strategies for antibody-drug conjugates. Here is an outline of today's topic. We will start from an overview and challenges of ADC CMC, and followed by discussion on the considerations for ADC process optimization. And in the last section, I will go through a brief introduction of ProBio CMC platform and a couple of case studies for process and analytical optimization. ADC is a novel therapeutic format and has great potential to make a paradigm shift in cancer chemotherapy. This new antibody-based molecular platform enables selective delivery of a potent cytotoxic payload to target cancer cells, resulting in improved efficacy, reduced systemic toxicity, and preferred PK/PD and biodistribution compared to traditional chemotherapy. The structure of ADCs can be divided into 4 major parts: the antibody, the payload, the linker, and the conjugation method. I think there was a previous question asking about the desirable features of the antibody. So briefly, it should have high affinity to the design target, high endocytosis, longer half-life, and low immunogenicity. For the payload, typically it should possess high cytotoxicity with high stability in vivo, and preferably strong transmembrane ability that enables bystander effect with well-known MOA and short half-life. For the linker side, it typically should be stable in plasma and allows drug release in the target cells. Ideally, it should be more hydrophilic and cleavable in vivo. And nowadays, there is a rapid advancement in the conjugation technologies, which allows more specific conjugation site, a desirable DAR value and distribution, and low heterogeneity. This slide summarizes an overview of current conjugation technologies. As we can see, there are some commonly used methods that are already available for clinical tested ADCs, such as lysine or cysteine-based conjugation. The advantage of those conjugation methods include simplicity, and it has been already approved and clinically tested for commercially available drugs. On the other hand, those conjugation methods typically gives either very broadly distributed DAR and heterogeneous products. Therefore, there's potential reduction of antigen binding or increased clearance with distribution of DAR. On the other hand, there are more and more targeted conjugation methods, such as engineered cysteines, introduction of non-nature amino acids, enzymatic conjugation, conjugation, etc. For those groups of conjugation methods, the biggest advantage is more defined DAR and a more homogeneous products. However, the majority of these methods requires genetic engineering of the antibody, therefore typically result in potential immunogenicity issue or result in lower titer of expression in the production process. And for some methods, there are special agents or enzymes involved in the process, which will further complicate the production process and increase the overall cost of the manufacturing process. And other methods include cysteine rebridging with a highly defined DAR and more high structure of stability. However, it also could potentially involve in disulfide scrambling problem, etc. Due to the complex and heterogeneous properties of ADC, a lot of challenges are posed for ADC CMC development. Here I listed a number of key concerns, starting with some safety concerns due to the handling of high potent or toxic compounds. Therefore, a dedicated lab or facility with special EHS requirements is typically needed, and cross-contamination control and requires experienced staff for the development and manufacturing process. And for the conjugation process, it typically introduces more complexity in the manufacturing process, including the control of drug load and heterogeneity, the free drug contents, the residual solvent control, high molecular weights and other impurities, stability of intermediates, and a lot of considerations for scalable process scale. On the other hand, due to the complexity of the ADC drug, which involves multiple components with very different physical and chemical properties, that pose a lot of challenge for analytical method development, method qualification, and process characterization. So we will have a further discussion on these points in the later section. And also the formulation and purification process, because ADC is typically unstable and prone to form aggregation, which often requires special formulation or lyophilization process. And last but not least, that introduced a lot of complexity in the supply chain process, because multiple manufacturing facilities are going to be needed to produce both the naked antibody intermediates as well as linker payload, the conjugation of the ADC, and the final finish and fill for the ADC DP. There is currently a lot of discussions regarding the manufacturing of ADC on different sites, and of course that pose some additional challenges for the overall supply chain management. In this section, we are going to go through all the life cycles for the ADC process development and manufacturing, and go over some key considerations for each step. We will start with some considerations for the naked antibody manufacturing. The quality attributes of naked antibody can have great potential impact on the ADC final product. And that include some direct impact, such as the residual impurities that will be carried over in the ADC DP, and indirect impact, such as residual impurities that may affect the conjugation process. And therefore, the spec of naked intermediates and some specs need to be strictly controlled, such as the high molecular weights and the mismatch of disulfide bonds, especially for some bispecific and multispecific antibody. The trisulfide bond and free sulfhydryl contents, which may affect the cysteine-based conjugation, the C-terminal lysine modification, which may affect the lysine-based conjugation, the glycosylation profile that may affect some glycol-based conjugation, and special amino acid for some site-specific conjugation. As you can all see, those are the key specs that may have some potential impact on the further conjugation steps. On the other hand, some other parameters, such as antibody concentration, pH, or conductivity requirements may be relaxed because we are going to go through buffer exchange and further conjugation process that may change the original parameters of the naked antibody intermediates. And the buffer system should also be considered to be comparable with our further conjugation system. Some unsuitable components of pH, such as some buffers with free amino groups that may react with lysine conjugation system, need to be buffer exchanged before going through the next conjugation step. For the linker payload, special attention should be paid to the production process, which should be robust during scale up and consistent among batches. So if you purchase some linker payload from external vendors, you should also pay attention to the difference between GMP versus non-GMP quality standards for those linker payload products. And the linker payload should keep stable during storage and transportation until the conjugation step. And typically, sometimes it requires special storage conditions in order to ensure the stability of the linker payload species. Regarding the spec of linker payload, similar principles should be followed. So those parameters that should be strictly controlled are those impurity and attributes that could potentially impact the further conjugation process, especially some conjugatable impurities that can hardly be removed after conjugation, such as some isomers of the small molecules. On the other hand, some other parameters, such as non-conjugatable impurities, which can be removed after conjugation, and residual solvents, such as some organic solvent used in the small molecule production, because some of them will also be needed in the conjugation system for the linker payload dissolve step. Those requirements might be relaxed. Regarding the conjugation, there are a number of key considerations in this particular step. Starting with the selection of solvent and material addition, because the first step is to dissolve the linker payload into typically organic solvent. The selection of solvent typically requires high solubility for the linker payload. It should be miscible with water, protein friendly and inert. A number of examples include DMC, DMSO, DMA, DMF, etc. And during the addition of the small molecules, it typically should be based on weight instead of volume, and some attention should be paid during the experiment handling, such as flushing the containers, and also the purity of the reagent should be considered, and appropriate addition sequence should be followed in order to have a very accurate control of the reagents. And the most important aspect is the control of the DAR and distribution to enable very precise control of conjugation conditions. A number of key parameters such as the buffer system and pH, the conjugation time, how to control the reactions, accurately control the reaction start and quench, conjugation temperature, and also special consideration should be paid during scale up as well, in order to have precise control of those key conditions. And also to minimize off-target conjugation, here I list a number of examples. For lysine-based conjugation, we should normally avoid non-specific conjugation to other amino acids such as serine, tryptophan, which contain free amino groups that is easy to hydrolyze and affect the DAR and overall safety of the final product. For cysteine-based conjugation, excessive MC may react with the residual lysine and causing an increase in the desirable DAR. This could be controlled by optimizing the conjugation pH. And last but not least, we should optimize the process to minimize potential product aggregation. There are two groups of typically product aggregates, the covalent-based versus non-covalent-based aggregates. So there are a number of conditions that we could potentially optimize to minimize those aggregation formation. Typically, to control the covalent-based aggregates, we should have better control of the reaction conditions, including lower protein concentration, lower reaction pH, and the overall storage time. And to control the non-covalent aggregates formation, typically we should consider to reduce the hydrophobicity of the drug, control the reaction temperature, the mixing speed, and hence the shear force induced, and control the solvent's concentration. On the analytics side, here I list a summary of the key quality attributes that should be specifically developed for the ADC. Here I didn't include the common analytical method for naked antibodies because essentially they are very similar. So for conjugated drugs, the DAR value and drug load distribution, the methods typically include for cysteine-based conjugates, HIC-based HPLC native mass spec, RP-HPLC, and for license, due to the higher heterogeneous generality, UVVs or LC mass might be more appropriate methods. And similarly, for site-specific conjugation, using HIC or RP-based HPLC, LC mass, or native mass are good methods to quantify the DAR. For the conjugation methods, most commonly used method is LC mass spec method, and free drug to develop RP-based HPLC or LC-QQQ mass spec. To quantify residual solvents, the GC or RP HPLC methods is commonly used. And again, methods for heavy metals include ICP-MS, and also biological assays are needed to specifically analyze some biological properties such as the cell-based potency assays, bystander effect, and payload MOA-based effect. So in GenScript, we have our established internal cell banks to support the majority of these bioassays. Moving on to the next consideration is the safety control, as this is one of the key challenge for the potent drugs. Here, during the entire production cycle, different attentions should be paid and for the different process and at different stage of the CMC development. Typically, a hazard assessment is used to determine the OEL levels and respective OEB needed to apply site-specific handling and safety measures. For us, since we are CDMO, potentially handle a range of different potent drugs, so we apply OEB5 level safety requirements for our facility. And in terms of the process, there are a number of key processes during the ADC manufacturing, starting with antibody modification, followed by conjugation and purification step. And the last step is typically the formulation step. And in the process development lab or process development stage, during the antibody modification step, typically this is non-toxic, so only requires standard gowning, and the lab requirement is typically GLP standard. And the conjugation is the most critical step, which involves the dissolution of the powder payload into the solution, and therefore requires highest attention for the safety purpose. Therefore, OEB5 level isolator and protection shield and laminar flow cabinet are typically equipped in the conjugation and following purification lab. And in the formulation stage, since most of the toxic agents have been conjugated to the antibody, and basically that involves a buffer and excipients addition, we have the OEB5 still the isolator and protection shield required in this lab. For the clinical and commercial GMP manufacturing process, again, for the antibody step, typically it only requires a standard gowning for the GMP clean area requirements. And moving on to the conjugation stage, the high safety requirements is applied, including OEB5 isolator, special laminar flow cabinet, and special protection equipment for the high potent API process. And the formulation room requirement is similar, followed by the isolator and appropriate PPE. And there is also special requirements regarding the facility design, including differential negative air pressure independent exhaust system with HEPA, bag-in, bag-out system, function segregation, fog shower room, etc. The last part is regarding the scale-up process for ADC, CMC. So here I list a typical scale considerations, starting from the lab scale process development to pilot scale to commercial scale production. And some attention should be paid when scaling up the equipment, including the limitation of the control of the equipment, such as the accuracy of the temperature, pH control, the feeding speed, the pump speed, the stirring limit of the equipment. And nowadays, typically, there are two different systems, either based in stainless steel equipment or disposable equipment. So for stainless steel equipment, cleaning verification is required, especially for toxic payload. Therefore, even small amounts of residues can pose some safety risk. Therefore, a very highly sensitive quantitative method is required for those type of facilities. On the other hand, you can choose to use disposable equipment. Therefore, no cleaning validation is required, but material compatibility and leachable studies are required because organic solvents is typically used during the production process. Again, the proteins amount should be accurately controlled, considering the density of the recovery solution versus the stop solution, and good operability. Facility should be considered for conjugation and purification process, including a good control of the CPP and KPP of the process. And if there's potential changes of the antibody post-translational modification or conjugatable impurities, appropriate comparable studies should be conducted. And last one, we just discussed the safety controls should be designed based on OEB or OER level. of your product toxicity. Okay, let's move on to the last section on the introduction of our platform and a number of case studies for process optimization. Here is an overview of the ProBio one-stop ADC platform. Here I will focus on the CMC part. On the antibody CMC, we have a wealth of experience focusing on different molecular types, including monoclonal antibody, bispecific antibody, and antibody fragments. We offer fast CMC service. The sequence to docs batch can be as fast as 6 months for monoclonal antibody case. And we have so far a track record of 5 IND clearance of ADC naked antibody CMC projects with 2 projects ongoing. And on the newly developed conjugation process and formulation development, we have established in-house template process for licensed cysteine-based conjugation process and a field map and enzyme-assisted conjugation for site-specific conjugation methods. There has been established process to better control the DAR and aggregation and establish formulation methods, including lyophilization. In terms of track record, so far we have obtained 3 IND clearance of AXC CMC projects with 2 projects ongoing. Again, we have well-established analytical methods, which covers the physiochemical, biochemical, biological methods to determine the DAR, residue solvent, and cytotoxicity. And last but not least, our recent established manufacturing capability include both DS and DP GMP facility in Suzhou, which include 50 to 200 liter scale conjugation bioreactor and annual capacity of 30 batches per year, which we'll go through some details in the later slides. In terms of the timeline, our team is able to support 9 months from DNA synthesis of the naked antibody to ADC tox batch. And the overall timeline is shown in this slide. As you can see, this is from the DNA synthesis of the naked antibody all the way to the GMP production, including the DP production of the final conjugated ADC products. And of course, we can offer more flexible service covering different stages of depending on your needs, including naked antibody development, manufacturing, conjugation, and following formulation development, and also the stability studies covering both DSI and DS for the ADC product. Here is an overview of our bioconjugation process development lab. Here I wouldn't go through this word by word. Overall, this shows the bioconjugation process development facilities and capabilities. We adopt both OFAT and DOE approach, which we'll go through some details later, and some platform-based process for licensed cysteine-based conjugation, and of course, post-conjugation purification process, including different modes of chromatography-based purification and UF-DF process. For the formulation, we cover end-to-end formulation process for bioconjugated products, including lyophilization process development and further stability study and in-use study as well. And in general, our PD lab covers the scale down to 1 to 2 ml of the small scale optimization and up to 50 to 10,000 milliliter volume in our PD facility. And regarding analytical development, we have physiochemical properties, mostly HPLC-based and mass spec-based methods to determine DAR, primary structure, and drug conjugation site and purity levels. For the biological properties, for lot release, we adopt the binding potency, ELISA-based binding potency, and cell-based cytotoxicity potency. And for characterization, the FC affinity and FC function assays, and we can conduct those studies to compare naked map versus ADC to evaluate the influence of conjugation, bystander effect, and payload MOA-based effect bioassay. And here is an overview of the drug substance, GMP production, and fill and finish DP facility, and with the parameters and capabilities listed in this table. If you have any further questions, feel free to reach out to myself or our BD team for further information. And here I'd like also to mention that with the introduction of the antibody class concept, and more and more drug developers are paying attention to AXCs beyond the ADC concept, which basically involves a variety of different antibody formats or protein formats, as well as the drug formats. For example, in our platform, we have worked with a number of clients on the ARC, which means antibody radionuclide conjugation CMC project, and we have one currently ongoing, which will file EMA and FDA by the end of this year. And we also provide a one-stop AOC CMC, which means the conjugation between antibody and oligonuclease development, which is in collaboration with the broader GenScript capability, the GMP siRNA enabling service. And of course, there are multiple targeting molecular types, not limited to naked antibodies such as bispecific antibody fragments, and proteins or peptides, etc. So far, our platform has successfully delivered 3 this type of CMC projects. And since our ADC capability is new, we haven't worked directly with ADC drug conjugates. However, we have in the past worked with some AXC conjugation projects with a very similar conjugation mechanism. All right, we will move on to a number of case studies to demonstrate how we do process optimization. So in this slide, this shows an overall process flow for ADC manufacturing. And we will focus our attention on the conjugation part following the production of DSI. So typically it goes through, if needed, antibody modification could be applied, such as reduction, oxidation, or introduction of some site-specific process, followed by a buffer exchange if needed. And of course, the conjugation process with a control of number of key parameters, the quench, following by the purification process, which involves various modes of chromatography if needed, and of course UFDF to remove the small molecule drugs as well as free residue solvents, etc., and formulation. Regarding the conjugation process, the first case study is one ARC conjugation we recently completed development. So we start with, again, the manufacturing. This is 80-liter GMP DSI production with the summarized quality attributes. And after that, since the purpose of this project is to conjugate radioisotopes for ARC treatment. Therefore, on our side, we perform a lysine-based conjugation process with the delta SCN linker chelator. So the amino group react with SCN. This is a typical lysine-based random conjugation process. And the delta group will be performing the radiolabeling step on site, typically in the hospital. So we developed a platform process based on this project, and we start with an optimization of a number of factors, such as how to effectively quench the process using low pH. We conduct a conjugation time cost study and find out the reaction efficiency decrease with the increased reaction time. However, there's a highly linear correlation with the reaction time during the early stage of reaction. And we conduct OFAT studies for a number of key factors, including the molar ratio, pH, temperature, the shifting speed, and DSI concentration. To establish a more well-controlled process, we leveraging this study, we conducted a more detailed DOE study looking at a number of key factors that might potentially interact with each other, which include DSI concentration, the molar ratio, and pH. By performing a well-designed DOE study, we identified quantitatively the potential impact of these different factors on the reaction DAR. Therefore, we come up with a quantitative model to predict the DAR distribution for different conditions within our design space, as shown here. And later, we performed a number of experiments utilizing different parameters within our design space and validate our design model, as shown here. Even we adopt different conditions, reaction conditions, the predicted DAR by the model is very close to the final DAR by our experiments, with the deviation for most cases within 10%. So this is essentially our approach to adopt most of this random conjugation, because in the manufacturing process, you should have a very good design space and process understanding to accurately predict potential process deviation. And after that, we did a comprehensive robustness studies covering our design space and successfully showed a process scale-up process from the small scale 1 mil all the way to 25 mil, 400 mil, and 20 liter final DS production stage, with a DAR value very close to our target DAR of 1.8, and very close distribution of the DAR as well. The next case study is a pretty complex three-step conjugation, which involves conjugation of two antibody fragments, which is single-chain FV. So here I showed the basic mechanism of this particular case. So under reaction one, so this is, again, even though it's slightly different from the ADC case, the underlying mechanism is particularly similar. So for the first conjugation step, it involves reduction of disulfide bond on the C-terminal, and it reacts with maleimide and to generate the single-chain FV with linker 1. And for the second fragment, it reacts with two linkers with the reduction of the disulfide bond with reaction with maleimide on one side, and on the other side, the amino group react with NHS groups to form the fully linked linker, leaving azide groups to react with DBCO groups as a final click chemistry conjugation to generate the final product. As you can see, this is a pretty complex case, which involves first a good control of substrate purity to avoid any byproducts during the first two coupling reactions, as well as the last the conjugation reaction. And we need to control the multi-step purity of the reagents in order to maximize the conjugation reaction, and also to remove undesirable impurities after the conjugation process. And we have completed this whole project for a 50-liter scale GMP production and successfully filed IND for FDA for this particular project. So the next couple of slides is just to show how we can optimize the purification and conjugation process to maximize the reaction efficiency. So first, as I mentioned, single-chain Fe is not a very stable fragment, so we need to control the substrate purity to avoid byproducts during the conjugation step. So here it shows how we purify the reaction number 1 and the reaction number 2 to significantly increase the purity of the final product using CEX and AEX based chromatography. And here briefly showed a control of multi-step conjugation process, because essentially there are 4 of 3 conjugation steps ongoing in this whole process. So reaction involved including the reduction of disulfide bonds, reaction of amino plus NHS ester, sulfhydryl plus maleimide, and click chemistry. So we explore the optimization process for the different reduction and conjugation conditions and identify the optimal reaction conditions for this different process based on different mechanisms. So for this case, for example, it shows the ratio of the, for the final conjugation step, the ratio of the single-chain Fe1 versus 2 is essential to improve the overall reaction efficiency. And last slide, it shows here is, again, the purification process for the final product, because the reaction efficiency is not that particularly high. So a lot of impurities, especially high molecular weight and low molecular weight species are present. So we designed a series of polishing chromatography steps to remove undesirable side products to ensure the final high purity of the final product. And regarding the optimization of analytics, here, the first case I'd like to discuss here is on the ADC concentration determination, because some payload linkers may have some interference on the solvents at A280. For example, for this particular case, for this drug linker, the maximum absorption happens at 270. However, it also has some absorption at 280. So that introduces some interference when you try to calculate the antibody concentration using UV-based absorption model. So typically a correction should be performed to better predict the final antibody and ADC concentration. So here is sort of a universal method. So you should look at the absorption at 280 and absorption at particular, for this case, 270 regarding both the drug species and mAb species. So combining the equation 1 and 2, you can obtain the ADC and antibody concentration. So here shows an example of the difference if you didn't do the correction. So you might result in over 10% deviation in your predicted antibody concentration. And here is an illustration of using LC-mass method to quantify the DAR distribution. For example, in our experience for the typical lysine-based conjugation, because the product is highly heterogeneous with respect to both the DAR as well as the conjugation site. So typically this is maybe not the best methods to quantify the DAR distribution because all the different peaks, even after separation, are still part of the mixture. Instead, using LCMF methods can be maybe more accurate methods to measure the DAR value distribution and measure the change of the molecular weight after conjugation. And you can follow the intact mass spec methods to accurately quantify the DAR and DAR distribution. With that, I think because of the time limit, I didn't have time to go through a lot of case studies, but I hope I've able to successfully demonstrate that the complexity of the ADC CMC process, as well as our internal capability and experience with respect to some conjugation case. With that, I'm happy to take any questions you may have, and thank you very much for your time.
Thank you. Thanks for Dr. Li's excellent presentation. Okay, let's move to the Q&A session. If you have any questions, please type it on the chat or Q&A panel. As you enter questions, please also follow our LinkedIn account, GenScript profile, and Korean audience can also scan this QR code and follow our Kakao channel. There will be more online resources, and we will share them on our LinkedIn and Kakao channel at the first time. Thank you. Hi, Dr. Li. We received a question from the audience. It's a pretty long question. Could you please check it in the chat room?
All right. Are you referring to the question you typed in the team message, right?
Yeah, okay. You can start with that too.
Okay. I will read out the question for the rest of the audience. Let me see. As you mentioned, we have to identify the conjugation site. Do we need to do the same for other product types such as AAV? I'm not sure because in our experience, we haven't done the conjugation with viral vectors such as AAV. So I think this is a very interesting question. But I imagine regardless of the targeting molecule you use, for example, we discussed different type of antibodies, identification of conjugation site, typically based on LC mass spec method, is pretty critical for the drug release purpose. Hope that addressed your question. Thank you. And the next question is, simple bioconjugation using lysine or cysteine can lead to variability in DAR, but site-specific conjugation requires significant engineering of the map, which can give a better control over DAR, which is correct. Can you please provide some perspective on when would you select which of these two approaches? And in your experience, what is the view of regulatory authorities when it comes to the approval with regards to one of these two approaches? Thank you. Okay. I think this might be a good question for Dr. Chen as well. But I will provide my view here. I think in the past, as you can see, all the approved ADC drugs have been utilizing either license or system-based conjugation. This is partly first due to, I think, because advancement and development of the site-specific conjugation is only approaching in the recent years. So I think it also has to do with the maturity of different technologies because of the simplicity of the license of system-based conjugation. It is actually more commonly used in the approved drugs so far. However, I think in the longer term, as we can see, more and more clients in the ADC specialized field tend to choose site-specific conjugation method because it has much better control of the heterogeneity of the drug, therefore much better control of the efficacy and therapeutic window and the overall product quality profile as a result. So I think, in my view, in the longer term, we will hopefully soon see some approved drugs using site-specific methods because of the above mentioned advantages. However, as you correctly mentioned, there are some CMC challenges. I think I have discussed this briefly in today's talk. For example, a lot of SSC methods involve the use of enzyme. Some challenges needs to be certainly overcome when it comes to, for example, the production of those enzymes, the utilization, and the residue detection, etc. And of course, the overall higher cost of the manufacturing. But in my view, I think those are challenges that we could potentially, with the development of CMC and more experience, we could certainly overcome the majority of this challenge in our development and manufacturing process.
Okay.
And hopefully that addressed the question.
Okay, thanks for Dr. Li's sharing. We also received some questions during the registration. Let's take a look at the first question is, could you please kindly share us the general commercial manufacturing scale for adoption of anti-cancer and ADC?
All right. I think I partially covered this question during my talk. For the commercial, typically clinical or commercial stage GMP production, it involves a few hundreds, like 50 to 200 up to 500 liter scale manufacturing process in order to supply a manufacturing a commercial products. Again, it's up to our experience so far, again, with the further development of more ADC candidates and depending on the potency and efficacy of their drugs, maybe different scales might be required, but this is the current most commonly required manufacturing scale we have seen so far.
Okay, thank you. The next question is how to manage the OEB or OEL requirements across the ADC production cycle.
Right. I think I actually specifically designed a slide to address this question, if you could recall. So there is one slide covering the different OEB, OEL requirements as well as the safety protection requirements for different stages, covering conjugation, antibody stage, antibody preparation stage, conjugation stage, and formulation stage. And I compare the difference between the PD lab versus GMP manufacturing lab. I hope that helps address this question. And if you still have questions, happy to chat offline later.
Yeah, okay. Next question is non-antibody scaffold protein binder drug conjugate CDMO also possible?
Right. Yes, the answer is yes. So we offer, as I mentioned in one of the slides, we offer AXC type of extension in addition to the conventional ADC service. So again, regarding that that covers a variety of different antibody, like different types of antibodies, as well as potentially peptide or protein-based conjugation methods. We're happy to chat with you on your detailed requirements. For some process, such as, for example, the peptide-based process, we may need to extend our collaboration with the GenScript group, because sometimes the production of the peptide involves the microbial fermentation, such as E. coli system, etc. So we might leverage the capability of the GenScript group on the GMP manufacturing of peptide. For the other different type of proteins, such as the fusion proteins or recombinant proteins, if you have a desirable sequence that can be manufactured through the traditional recombinant control system. So definitely we can handle those cases, covering from the protein manufacturing to the conjugation process. Happy to take the request as needed.
Thank you. Here is a new question come from the audience. I also sent it in the group. Do you analyze the all residue enzymes used in conjugate? How much of their enzyme should be reduced? Is there criteria?
That is a good question. So far, we haven't worked on the GMP manufacturing using an enzyme-based conjugation yet. So, but if we have in the future the GMP manufacturing process for the residue enzymes, I think definitely the residues should be analyzed. All the residue enzymes should be analyzed for the final DS and DP products, and how much of their enzymes should be reduced. I think it depends on the specific reaction. However, regarding the specific regulatory requirement, I need to check out later, but I would imagine that the question is, what is the level of residue enzymes should we keep in the final product? So I will check with our regulatory experts and analytical experts to address that question regarding the residue enzyme level in the final products. Thank you. So I would imagine there is a criteria, even though this might be an emerging field, but there might be some criteria posed, for example, similar to the residual level of HCPs, I would imagine. So typically for antibodies, less than 100 ppm, for example. But I need to check regarding specifically the residual level for enzymes used in the site-specific conjugation process. Thank you for the question.
Yeah, thank you. The next question is there any Korean agent? How far do you cover ADC director users?
The answer is yes. We, of course, ProBio has worked with a number of Korean clients in the past, covering like bispecific antibodies, monoclonal antibodies, etc. Since our ADC service is kind of new, we didn't work directly with the ADC client. However, if I remember correctly, we have worked with a few clients to generate naked antibody for their ADC process. And second, we have successfully achieved one of the IND approval for both FDA and Korea FDA with an AXC conjugation, which is antibody-antigen conjugation project, CMC project. And we look forward to work with more ADC or AXC clients in Korea. And I think so far we have a number of cases under discussion for potential CDMO or CMO collaboration. Thank you.
Yeah, thank you for your answers. That's all the questions we have for now. If you have any more questions, please kindly contact us through our official email. Let me join others in thanking Dr. Chen and Dr. Li for their time today. Thank you for attending our webinar. Look forward to meeting you again soon. Thank you.